Welcome everybody. On behalf of Scifest Africa, a warm welcome from me, Janice Limson, to the second in our Scifest Africa webinar series. The topic today is diagnostic tests, and in a COVID era, access to accurate diagnostic tests is a game changer for any nation. I'd now like to hand over to Steve Sherman, who will be our host. Over to you, Steve.
Thank you very much, Janice. We have Prof. Janice Limson. We have Dr. Manfred Scriber. Is that pronounced correctly?
Yeah, it's okay.
Good, I'm doing well. We have Prof. Kevin Land. We have Dr. Andrea Julsing. Is that correct?
Almost, you'll sing.
You'll sing, and you'll certainly sing when we get to ask you questions. And then, of course, we have Tamlyn Shaw, and we have myself, Steve Sherman, and we have our Scifest support team. We're going to be just chatting about something that is incredibly topical right now. The COVID-19 pandemic has taken over everything that we do, and testing is critical. So what I'd like to do is ask our panelists if they can briefly introduce themselves so that we can find out a little bit more about who they are and what they do. And then I'm going to start hitting them with some questions. And if you've got questions, please send them through, especially our attendees. Type them in the chat. We would love to see if we can throw some of those questions at our panelists. So we'll start off with you, Professor Janice Limson. Would you like to tell us a little bit about yourself?
Well, I'm Janice Limson, Professor of Biotechnology at Rhodes. And it's been a real pleasure being a part of this specific webinar, as my field of research is in diagnostic tests. And we are specifically interested in developing low-cost diagnostic tests. So based here at Rhodes University, we have a special facility called the Biotechnology Innovation Center, which is where I'm based. And here we do, our research is, interestingly enough, involves not just the research and the innovation component for diagnostic sensors, but we're also heavily involved in the engagement aspect, as we feel that that's quite an important part if we're going to be developing tests that actually go into commercial reality. So we believe bringing the public with us is quite important. So for me, this webinar is quite special, and thanks very much to the rest of our panelists for accepting our invitation to join in.
Wonderful. We'll go to you, Dr. Manfred Scriber. You're going to tell us a little bit about yourself.
Yeah, thank you. So I'm from the Center for Nanostructures and Advanced Materials at CSIR. And I'm a manager here for a group that develops and scales up nanoparticles and also nanocomposites or plastics. And there's also an interest, and together with Professor Kevin Land, I'm managing a project for the Department of Science and Innovation that looks at putting together facilities that will assist with making these diagnostic kits and also printed electronics and so on. So I'll talk a bit about that later. But my background is quite diverse, from electronics to mechatronics, image processing, and also material sciences and nano. So that's me.
Well, I'm sure there's a lot more to it, but you've been very gracious in keeping your biography quite short. Let's go to Dr. Andrea Julsing. Would you like to tell us a little bit about yourself?
Thanks so much. So yes, I'm Andrea Julsing, and I'm from SAHPRA, which is the South African Health Products Regulatory Authority. And traditionally, SAHPRA, it was called the Medicines Control Council, and we would regulate all the medicines that you see on the market. Now, since 2017, we changed to become SAHPRA, and we're responsible for regulating medical devices. And so when we look at these type of diagnostics and all the tests that are being used, and in fact, there's quite a number of medical devices and IVDs that are being used now during COVID, and SAHPRA is responsible for regulating those and ensuring that the products that are made available are safe to use, they perform as they intended to perform, and that they're of good quality. And just as a background, I'm a pharmacist, graduated from Rhodes University, so I'm delighted to be part of the Scifest program this year. And yeah, so I've been with the regulator for I think over 8 years now, and prior to that I was involved in pharmaceutical production and production of complementary medicines, and very excited to be part of the medical device regulatory framework now as it unfolds in South Africa. Thanks.
Very cool. And then, of course, we have Tamlyn Shaw.
Hi, everyone. Yeah, I'm also very excited to be part of this webinar. So I've got a master's in biochemistry from UCT, and I'm currently one of the co-founders and directors at Bio Pharms, which is an exciting new spin-off company from UCT that's focused on producing recombinant proteins, antigens and antibodies in the plant-based expression.
I think you might have muted yourself at the last bit there.
Sorry about that. Yes, yeah. And we're currently focused on producing corona-specific antigens and antibodies specifically for the serology-based rapid diagnostic test kits. And we're working very closely with 3 local test kit manufacturers to develop these tests.
Wonderful. Okay. And then we're going to go to Professor Kevin Land.
Hi, yes, thanks, Steve, for the opportunity. I'm currently based at the University of Pretoria in a part-time position, and I also work for myself. I was previously with the CSIR for quite a long time, where I established the microsystems capability at the CSIR. So really what I'm passionate about is the whole area of microsystems, whether that's from the micromanufacturing perspective of manufacturing really, really small things, or one of my real passions is microfluidics. So the study of and control of very, very small volumes of fluid, from femtoliter, nanoliter, those kind of volumes of fluid. And that has specific application for all sorts of diagnostic tests and applications, and also looking at different platforms on which one can develop these different tests, whether it's an equipment-based platform or whether it's equipment-free platforms, which would lead to low-cost diagnostics, which Janice was also mentioning. So that's kind of my real passion at the moment.
Awesome, awesome. Well, we've got a very well, what's the word I'm looking for? Highly esteemed panel of people who actually know what they are talking about. I did notice that a few of you might have thrown in a few terms which went over my head, and I'm just going to play the role of the average person listening to this webinar who might not understand what some of those terms mean. So we will certainly interrupt you and ask you to clarify or explain what some of those terms mean. I think the best way to start is to perhaps let's do a quick round of questions to each of the panelists, so we can just warm you up and get you comfortable to answer some of the questions. And I will start with Prof Julsing, Dr. Andrea Julsing. We're going to hit you first. We're going to go with, if I go to my doctor and I need to, or maybe not even to my doctor, to a lab, if I need to have a test for COVID-19, what tests are available right now in South Africa? And maybe you can also just touch on, you know, are these tests quick to do? What do they cost? Would you be able to address those issues?
Yes, perfect. So, and actually I believe these days you can even go through a drive-through to have a test done. But what's available at the moment is a molecular test or a PCR test. This is a test that actually detects the genetic material of the virus. So you would go to your doctor or go to the drive-through, and they would then do a swab of either your nose or your throat. And I actually haven't had a test done myself, but I believe it's quite uncomfortable because they need to go quite deep, you know, into the nasopharyngeal space. So it's where the back of your nose meets your throat. So it's quite in there. Then they take that swab and process it at the laboratory. So you cannot obviously do this test at home. You can't do this test at the pharmacy or at your local GP office. This test is processed by a lab. Now, also the technology has really advanced, and so we see for particularly COVID products, there were tests that took about 48 hours to process, but there are now also rapid molecular tests that can have a turnaround time of 2 hours or 3 hours, so it's much quicker. But what we do find in terms of, you know, supply and demand and shortages, often in the media they say we've got a backlog of tests and so on. It's not because we've got a shortage of PCR tests, but rather we see globally there's a shortage of the extraction kits. So first we extract and then we run the PCR. And so that's really where the backlog is starting. In terms of cost, so SAHPRA is actually not responsible for pronouncing on cost and so on, but just in our engagements with industry, you know, we've seen prices that could be from about 1,200 Rand and then dropping as more tests become available to about 700 Rand so per test. But yeah, I don't have firm figures in terms of costs and so on. But today, if you had to do a test, it would be the PCR test, and this would be to diagnose you and say, yes, you have got COVID-19.
Okay, all right. So let's stick on that topic a little bit. We're going to go to Prof Lan. I wanted to ask you, in general, how do the diagnostic tests work? Because there was mention of this PCR test. Maybe you want to also explain, you know, what a PCR test is and how it works. And then how do these differ from the antigen tests or the tests for antibodies? Can you maybe explain that a little bit for the rest of us here?
Sure. So the PCR test, which Andrea spoke about, that stands for polymerase chain reaction. And typically it's looking at the genetic material, so that's RNA or DNA in the sample. In the case of COVID, it's RNA. And it goes through this process, which is called the PCR, which goes through a thermal cycling process, which in very basic terms, it takes the DNA and it amplifies it. So there's, instead of one piece of the DNA, you can actually, within a fairly limited period of time, cycle through millions of times to get a lot of copies of the DNA. And if one gets then sufficient copies of the DNA, one can then obviously measure it. And that's why this specific test can be so sensitive, because it amplifies the material that you're trying to measure. On the flip side, you get the test, the antigen test that you're talking about. And just for clarity, typically one would implement this on what's called a lateral flow strip. And you can think of that along the lines of a pregnancy test. So if you test positive, you would see a red line on the test. And if you test negative, then the red line would not appear. So it's a fairly robust test, at least for the purposes of a pregnancy test. When it comes to COVID, like we're trying to do now, it's not as sensitive, as sensitive as the pregnancy test, because no amplification actually happens on the test itself. It directly measures the antigen in the sample that you're extracting. There is another lateral flow, which is an antibody test, which can also then measure, but it doesn't measure the actual viral load. It measures later. So it's the body's response to the virus. The body then generates antibodies, and those antibodies can then be used to tell whether you've actually had COVID or not. But it cannot be used for immediate diagnosis. So those are the two main tests that are used. The antibody tests can also be in other formats. So they can work in formats such as an ELISA test, which is small wells of fluid which one can test in a laboratory setting, or it could be an electrochemical test, etc. But the really main test is the lateral flow test that most people refer to when one's talking about the antigen or antibody test.
And if I understand correctly, and just for those who are trying to work through this with me, we have the one that can determine instantly that you have COVID-19, and that would be the PCR test. And then we have the other test, which tests that you've actually had it because your body starts fighting COVID and produces antibodies, and then we measure the antibodies. So that's post-COVID. Is that correct?
That's correct, but there is also a lateral flow test that can measure the antigen itself. So it would perform the same function as the PCR test, but these tests are not yet available in South Africa for use.
Aha, okay. Well, maybe they are, maybe they're not, I don't know. We're going to go over to Tamlyn, and I'd like to know, I mean, you're from the Cape Bio Pharms, and I want to know what part of the COVID-19 puzzle is your company addressing, and then maybe you could talk about the recombinant protein production facility, and you could talk about the reagents. What is that all about?
Okay, great. Yeah, well, thank you so much, Andrea and Kevin, for kind of setting the scene and explaining a lot of these concepts. So basically, the part of the puzzle that we are contributing to is we are making the proteins, and a recombinant protein is basically when you take the DNA from one organism and place it into another, and that other organism makes the protein for you. So in our case, the organism is a plant, and we take the gene sequence for the COVID antigen or an antibody, and we clone it, or we infect it into the plants, and then the plant recognizes that DNA and makes the corresponding protein. So in terms of the COVID response, we are making all the different antigens. So the spike proteins on the outside of the virus, the nucleoprotein on the inside of the virus, and different versions of those spikes. So we're making those antigens as well as antibodies that are needed on these rapid test kits that Prof Land was explaining. So the antibody test kits, where you put a drop of your blood on this kind of pregnancy test kit, and it flows over this membrane, and the antigens, those proteins in there, are conjugated to gold. And then you have what are called capture antibodies on that strip as well, which will then pick up these gold labeled antigens as well as the antibodies in your blood and form a line. And this can tell you kind of that you've had the infection and your body's produced antibodies against it. We're also making proteins for what Prof Land explained was the antigen rapid test kits. And we're actually working to use a method to amplify that signal. So yeah, that's basically us. We produce basically the raw ingredients that the test kit manufacturers then take and put on these test kits.
So, I mean, this is obviously a team effort, because you don't have one company specializing in its entirety. You've got to get the different components and put them together. And maybe that's where Dr. Scriber can come in. What I wanted to know from you is what sort of research capacity does South Africa have to develop new diagnostic tests? And the other thing is, what sort of capacity do we have to manufacture these sort of diagnostic tests? I mean, we can already see that in South Africa, we're about to hit that moment where the spike in our exponential graph is about to grow, because they've obviously lowered the lockdown level to level 3. And our expectation is that there is going to be a growth in the number of cases presenting themselves and possibly the number of deaths as well. So this is of paramount importance. This is when we need to know if we can get more tests and that sort of thing out there. So what is our capacity from a research point of view and from a production point of view? Are you able to unmute? There we go.
Yeah, somehow it was stuck. So yeah, that is a good question. And I think that question needs quite a bit of thinking. Firstly, one should not expect that research will immediately come to a device of which you can make millions. There are all the steps in the development process that you have to follow. And on the panel, we've got industry now, we've got SAHPRA, and we've got research. So all of those need to fit together so that you do the proper testing and validation and certification until it gets to the market. But the question was now on research capacity at first. And there also we need to distinguish between what we could maybe call the biotechnology or the chemical side, and then also the device manufacturing side. And here I would say that there is already research in place for the future, which will not just be the pregnancy type test we were talking about, but also incorporating electronics and the Internet of Things and so on. And in this regard, if we look already, then we've got quite a substantial research happening at universities. And I see Prof Janice Limson on my screen, who's already got one group. And we are actually consolidating this research into into nodes that will be funded by the Department of Science and Innovation to take the research to the next level. Also included there is the University of Pretoria for printed electronics, Stellenbosch University for biosensors, and also chemical sensors at University Western Cape, and also CSIR, which could look at lab on a chip. So things are happening, but we cannot expect them to just happen too quickly. However, we also need to mention that in the long run, we should not only look at now COVID or corona type viruses, we also need to look at TB and HIV, AIDS, and other ailments that we've got or pathogens that we need to detect. And all of those requires a proper research capacity, which we are actually supporting, as I said, through DSI investment. You were also asking about the capacity to manufacture. And already we've got Tamlyn looking at one aspect of it, and then there are other companies that already produce the pregnancy type lateral flow devices in this country. But we would say that the country has limited capacity. We need to change this. We should not just import all the time. We have to localize and we have to develop locally. And this is why Professor Kevin Land and myself are championing an initiative where we establish facilities that will be able to manufacture diagnostic devices, particularly for point of care. And this is an exciting development, and listeners need to watch this space, because at the moment we are in the process of finalizing our proposal with the DSI for substantial funding to do this, so that we can locally develop. And as I said, take devices and technologies to a level where we can manufacture them, so that we can take them out of the laboratory into the industrial environment. This is really crucial, and the Department of Science and Innovation is really forward-looking to invest in this. So that's really good. If we'd done this a couple of years ago, it would have been so nice, because the conversation now would be different. Then we would say, okay, this center has already produced so many thousands or millions. But COVID has caught us a bit too early.
Well, has it? I mean, you know, I've seen presentations on the internet, Bill Gates, Barack Obama. There were several people who spoke about a virus on its way. And that was a couple of years ago. So we knew this was going to happen at some point. We just didn't know when, but we knew it was a matter of, it was a case of it will happen, we just didn't know when. So why are we all so surprised that it actually did happen?
Well, I would say in this country, it's a matter of budgets and fiscal policy. There are so many needs that you decide from point to point what is most relevant at that point. So we've been busy with this for some time, and only now the budgets are available.
All right, well, I've got a whole bunch of questions that are coming through from the attendees. So I want to kickstart with one of them over here for Dr. Julsing. It says here that, would you be able to let us know how many coronavirus serology test kits have been currently licensed by SAHPRA, and what progress is being made in validating those kits for laboratory use? And that's from Chris Adams.
Great, thanks so much for the question. So we've actually received 205 applications for serology test kits. So that would be the finger stick test that could be used in the field as such. And out of those, we do a technical screen, we look at the data that's provided, and based on that, we would then channel any test kits that are looking promising in terms of performance to our national reference laboratories, so that the performance evaluation can be conducted. Based on the outcomes from the labs, it would then inform our decision making as to whether a test is performing adequately to allow it to be used widely in the country. So as we stand today, we've had 25 tests go through the laboratory. And I think the disappointing part is that the majority of them are not performing as we would expect them to, and also not performing as what was available and shown in the literature. So the challenge is that for many of these test kits, they work well in patients that are confirmed to be about 28 days post-exposure to the virus. So that's good, and that's a good place then to use the test. But it won't be useful then to have a test. If I just went out into the field today and just tested everybody, the test is not performing well enough. It's not sensitive enough to pick up on whether or not somebody's got the antibodies. It's not specific enough to say, okay, are these antibodies for COVID-19 or SARS-CoV-2 and not some or other SARS virus? So that's the challenge. And as we stand today, there's no serology test kit, finger stick test that is authorized for use by SAHPRA. And I think I should take the opportunity to say, if you are aware of anybody selling these test kits or advertising them on social media, or positioning them, saying that these tests are to be used at home, they are self-test kits, this is not the case. These test kits have to be used under the supervision of a healthcare professional. And so any reports should come straight to SAHPRA. We've got a regulatory compliance unit who would then be able to investigate any complaints.
Okay. Taryn, I think this question is for you from Kerry Gordon, and that is, are there any locally manufactured reagents currently being used? And what are the roadblocks to getting these made and approved for use?
So in terms of the recombinant reagents that we are currently making, we started off making reagents to sell to universities before COVID happened. And then luckily, because of the plant-based platform, I saw there was a question asking about the yields of recombinant proteins in this platform compared to the microbial systems. So the yields are quite comparable. The benefit of this platform is that you can pivot and change direction really quickly. It's kind of a plug and play. So we can make new proteins really quickly. So we've gone through kind of an iterative process with the local test kit manufacturers we've been working with to kind of supply them with samples of these proteins, and they then are feeding back to us which ones are working best, doing their own internal testing, and now they are ready to apply to SAHPRA for approval of their test kits. So it's been quite a long road over the last almost 3 months that we've been working closely with them to get to this point. And our proteins have been performing quite nicely, but they are only now being kind of applied for approval. So I'm not sure if that quite answers the question.
I think it certainly went enough of the distance to get there. What I want to know is from, in fact, this is not me, this is Stephen Lang. Hello, Stephen, I hope you're well. He wants to know what determines whether you should do a throat swab or a nasal swab, and why does it have to go in so deep? Is it not dangerous in untrained hands? I mean, are they taking a giant earbud and sticking that down your throat? Are they taking a curve? I mean, I've never gone for one of those tests. Have any of you actually been tested for COVID-19? None of the panelists. Oh, Janice has. Janice, we'd love to hear your comments. How deep did they stick that thing up your nose?
It's not very deep, Steve, at all. I mean, it's literally just in the back of your throat. And they didn't go through. No, thankfully, just in my throat. It's really
a throat swab, okay?
It's really not that deep, and it's not that long and scary. So.
Okay, but some of them are doing nasal and some of them are doing throat. What determines which method you should use?
I actually don't know the answer to that question. Andrea, is there anything from SAHPRA's side? Not really. I think it might just be personal preference of different laboratories, but I use one of the private laboratories. This is a couple of weeks ago when we all started feeling quite sick at home, and the test came back negative. Thank goodness.
Now, I had an earlier discussion with the panelists before we started and before we went live, and I knew this was going to come up. And that was, there have been a couple of issues about people getting false positives. can you explain what would cause a false positive? So, Kevin, would you like to handle this one?
Yeah, I can give it a go. I think there are probably two main reasons, and then the other panelists are welcome to come into it as well. One of the reasons could be contamination of the sample. And the other reason, and probably the main reason, would be that, and here I need to introduce a few terms, if you don't mind, Steve.
No,
when one talks about the accuracy of the test, one normally talks about the sensitivity of the test and the specificity of the test. The sensitivity of the test talks to the number of true positives, and really talking about how many false negatives one can expect to get. Now, a false negative is when you test negative, but you're actually positive. Okay. Now, the specificity of the test is talking to the number of false positives that one gets. Now, the main reason for this, I believe, is that something like COVID is a coronavirus, but there are many, many different types of coronaviruses. So if your sample has another type of coronavirus in it, for instance, then you would get a positive test result if your test is not specific enough for COVID. So you typically want your test to be 100% specific, but that's very, very difficult to achieve. So that means that many of these tests would pick up other coronaviruses, for instance, and give you a positive result when you're actually negative for COVID.
Okay, does anyone else want to add to that? I saw Tamlyn was doing a lot of head shaking, so I'm going to definitely ask her to comment on that.
Yes, no, I was just curious, Prof. Land, do you think it might be helpful in this case to kind of use PCR with serology testing in combination? Because if somebody tests positive using PCR for another coronavirus, and it could have possibly already kind of passed out of their system, and they've got the antibodies, and they possibly, so basically using the two together to try and get a clearer picture if you do get a false positive from one.
Yeah, I think there are a number of strategies that people have spoken about. The specific strategy that you're mentioning, one would probably have to look at the statistics behind it and see whether it's really useful or not, especially given that the PCR test on its own is giving probably the results that one wants. The different strategies that I've heard about are more to do with the actual antigen test and what happens if you, for instance, have a sensitivity of, say, 70% or 80 or 90%. How do you make this more accurate? And the theory is that because these tests in principle can actually be made really, really cheaply and very, very high volume, you can actually have multiple tests. So you could test today, tomorrow, and the next day, 3 tests in a row. And that would then obviously improve, you know, if each time you've got a 90% sensitivity, this actually eventually improves the end result of the test.
Okay. Dr. Scriber, I was thinking if we had to take COVID-19 now, does this offer us an opportunity to rethink or reshape the efforts to generate diagnostic tests and not just obviously COVID-19, but all types of diagnostic tests?
Well, that is a very good point. I would say a country, any country, should have a long-term strategy on this. You cannot just react to pathogens that come along and then think that quickly you'll do something. So there needs to be an accelerated investment in the research side, as I've said earlier, so that you are prepared for the future. And this is exactly what we've proposed, as I mentioned before, to the Department of Science and Innovation, and where the proposal is at the final stages, is to put together research and support nodes that will help with manufacturing devices for the future. And as I said before, it all comes together. The Department of Science and Innovation has invested handsomely in biosciences. They've invested in nanotechnology, and those two come together handsomely now, where you can produce these diagnostic devices and also printed electronics and the future type of things that come along. So your point is basically that we should, I think, prepare and have a good balance between the biotechnology side and also the device side, the detection, and also what Professor Kevin Land just said now, the cost aspect is crucial. We have to look at technologies that will drive the costs down so that these test kits can be distributed quickly to everybody. And that's a key aspect in also the spread of the virus is, of course, early detection at home or wherever you are at a low cost so that you can self-isolate and so stop the virus. So again, if we look at our approach, there will be 5 centers that will focus on different aspects of this. And I can't wait to get going with that. That will help tremendously.
What I find fascinating is that you've been discussing these things and planning these things for quite a while. Then COVID-19 hits. Then did you feel that the urgency was recognized a little bit more? Because all of a sudden people realize, okay, you make a valid point. We do need these tests out and we need to get this research done. Or was there already plans in place to put the funding in and get it supported by the government?
No, I must say in this case, the government is quite good. As I said, they are quite forward-looking. So already a couple of years ago, they identified this, what we call a nano and micro manufacturing facility, as an important aspect. And the issue of point-of-care diagnostics was central to that proposal from the beginning. So it's just a matter of how to budget and when to budget for it. So these development of test kits have been on the agenda for some time, and it's a matter of just waiting for the budget allocations, then it can move forward. So the DSI have been really clever in putting this on the agenda already quite a while ago.
Because now the funding will obviously come through and match that.
Well, we are hoping so. As I said, we're in the final stages of the proposal. So I hope somebody from DSI will listen and then take notice.
DSI, I hope you are listening. Dr. Julsing, in the news, South Africa has a large backlog of samples. Eastern Cape apparently has a backlog of about 20,000 samples. I don't know where they got the numbers from, but maybe that is the case. Do you think that such a backlog is only due to the shortage of reagents, or is the health department understaffed for lab techs?
Thanks so much. So SAHPRA can't pronounce on capacity within the Department of Health or at the NHLS, which is our National Health Laboratory Service. But what I can tell you is that the NHLS has been working very actively to increase capacity within their various laboratories. So they're also looking, a lot of new technology has become available in the space where we're able to process tests, many more tests per kit and per reaction as such. So you're able, instead of just doing 24 tests at the time, doing 96, or instead of running a test that takes 24 hours, it's now taking 3 hours. So they've put a lot of resource into building capacity in the labs to support, reduction or clearance of the backlog. So that is in place. And then also once we get either an antigen or a serology finger prick test product that can be made available and that performs well, it would also really support in clearing these types of backlogs, because you'd maybe be able, where we see now we've got a population or a cohort in the Eastern Cape, they've had the swab done, but the test hasn't been processed. We could then say this is a group where we could do an antibody test. If they test positive and if they're symptomatic, then we will confirm and do the PCR for them. But if they test negative, then we could say, okay, come back in 1 week, do the screening test again. So there are ways of, you know, trying to address the backlog. Certainly the NHLS has made, you know, great steps in terms of getting resources in and building capacity to address that backlog.
If we want to get more lab techs, this is for promoting STEM amongst the younger people. We want to get more people going into these sorts of fields. What would they need to be studying? Anyone want to answer that one?
I can perhaps answer. So they'd have to definitely do mathematics, which will make you very happy, Steve.
Very happy, like maths.
And science at school, definitely. And then coming to university, they can probably go through biochemistry as a route, and certainly chemistry as well. So I've looked at the list of attendees, so there are quite a few chemistry people there, and then definitely biotechnology would be a route. I had a question though, just to follow up to Dr. Julsing, just in terms of companies that have diagnostic tests and they want to go through the process of registering them. How long does that take? What does it entail? I mean, I understand that SAHPRA is obviously working very hard to try and move things along quite quickly in this particular case, but what's the general process to actually get a test approved at SAHPRA?
So our general process, and also, you know, with COVID coming along, a lot has changed, because typically we would process an application in about 2 months. That would be a normal kind of process. Now with COVID-related products, we're really striving to get a turnaround in 7 days. So largely we're able to achieve this for applications where, you know, everything that we required is submitted. So for a complete application, we're really moving quite fast. Now, where we've got a local manufacturer producing either a PCR test or a rapid test, what we've asked the local manufacturers to do is to engage with the National Reference Laboratory, who would then facilitate a validation study. So they will validate the performance of the test. And so when the local applicant applies to SAHPRA, they would then say, here's my application form for SAHPRA, here's the supportive document from the National Reference Laboratory, and then SAHPRA can review and based on the outcomes of that validation, that would largely inform the decision of whether or not to register or authorize the product for use. And that process, the labs, because I think what we saw at the beginning of COVID was a massive influx of imported products coming in. And then now it's come to our attention that there is a local manufacturers who've got products that are ready to go now, and they'd like to do the validation. And depending on the test kit, for a point of care serology test, you know, we've got already quite a bit of experience there. So that's taking about 2 days for the lab to actually do that performance evaluation. For a PCR test kit, that's taking a little bit longer. We're also busy trying to just streamline the coordination around that validation. And we've got a lot of support from the NICD, from the NHLS in doing that. So that's probably about a week or so turnaround. And now the antigen testing has just come to light. And so we're just building up capacity there to also do performance testing. And I believe they would be quite ready to go by the end of next week. And that should also, once it goes through, probably take about 2 days to get the report. So we're really striving to get a quick turnaround time and to facilitate local production and get good performing test kits out on the market.
Thank you very much. That's really encouraging to hear. And I think the one thing that we touched on earlier on was that this is not, that while this is great, you know, for COVID-19 testing, I think it also sets us up really well for the future in terms of being more agile in our responses, you know, in terms of validation, as well as, you know, going through the processes with SAHPRA. Thank you very much. Back to you, Steve.
Yeah, I mean, we could be hit by another coronavirus at any point. And it could be a different strain, it could be anything. And if we are not preparing for that already, I think that we're going to be stuck in the same position that we are now, where it's now landed in our laps and we have to try and solve the problem. But if we are preparing for that problem before it hits us, then obviously that will make life a little bit easier. There was an interesting question here from Chris Adams. He says, an article in Spotlight on the 8th of May by Dr. Tom Boyles from the Helen Joseph Hospital suggests that 5 to 10 patient samples could be pooled. So one PCR test could do 5 to 10 patient samples. Are there any comments on this approach? Anyone want to tackle this one? I see Kevin smiling.
I tell you what, I'll start the discussion and then everyone else can jump in. I think pooling is an accepted approach in certain circumstances. One has to just think about the complexities of it though, because the way it works is that you would get, I don't know, let's say 100 samples to test. And you split those samples into, say, 10 sets of 10 samples. So if one of those sets of 10 tests negative, then you know that all 10 are negative. All 10 of those samples in that pool are negative. But if it tests positive, then all 10 of those need to go into the next testing phase, and none of them can be rejected. So a lot of that depends, and there's a, you'll enjoy it, Steve. There's a mathematical approach to it.
Mathematics of this, I was just thinking now.
There's a mathematical approach to it, which also looks at the prevalence in the samples. What is your expected prevalence? So the lower your expected prevalence is, the more beneficial a pooled approach is, because the more times you will get a full set that's negative. Because if your prevalence is higher, and I'm talking now maybe 10% or 20%, then many of those pooled samples will come back with a positive test, and you anyway then need to retest.
Correct.
But it's a strategy that is worth looking at and does work. It is more complicated from a sample control perspective as well. So I think if you have automated ways of doing it, it's certainly worth it. If it's all manually dealing with the samples, then I think there could be some complexities in doing it.
I just think if you take a lab that is, short-staffed and you have to throw in complex testing with multiple samples, and I mean, you're asking for a higher probability of error somewhere along the line. But if it is automated, then obviously that won't be an issue.
Can I carry on?
Go for it.
I had my hand up just now, but no one saw it. So I wanted to talk just very briefly on two of the other points. One was what people should study. And I think it's important to say that besides the sciences, I think there's also a valid case to say that some of the engineering disciplines would also be very, very beneficial, especially when one talks about the device side of the equation. And in that respect, and hopefully we're going to address a lot of it in this proposal that we've put forward to the DSI, but there are a lot of very, very exciting technologies that are coming along. And some of them are not very new, but they are fairly new. And Manfred's talked about some of them, like printed electronics, but there are a whole lot of different technologies, like communication technologies, for instance. How do you get, if you're using a low-cost device like a lateral flow, how do you get the result to the centralized system where you can actually analyze results? How do you read the result? And also, you don't really want an expensive reader that also now requires maintenance, and it's expensive to purchase in the first place. So a lot of these new technologies like print electronics, you can print a communication device, for instance, an RFID or a near-field communication, a Bluetooth. You can print that onto the device and then communicate the data at very low cost to some central database. You can, for instance, print light sources onto the device, or you could print a detector onto the device. So you may get away from manual reading of the line, for instance, where there is a chance of user error. So you may be able to automate a lot of these processes. And Steve, you know as well as all of us, there's been a lot of talk of 4IR, for instance, the fourth industrial revolution. And I think what we're talking about here is there are so many aspects of it, because essentially lateral flow is a sensor. So if we've got millions and millions of these sensors, how do we get the data from them? How do we read the data? How do we send the data? And there are technologies that are coming to the fore where these things may be possible. And in that case, 4R is then no longer just a buzzword that everybody uses, but we can actually really, really practically implement it for something which would be very, very advantageous for South Africa, you know, in terms of locally developing something that makes real local impact for people in South Africa.
Well, I mean, I've already come up with a solution. What would happen if the test showed a barcode for positive or a barcode for negative, and you scan with your cell phone, boom, you can send the results straight away, low tech, but it works. Could that be possible?
There are people that have done that.
Sounds good. Yeah, I'd also like to chip in and say that David Jarvis from Lateral Labs has also been developing what's called MyPA, which is an app that kind of works like this, where the test kit would automatically send the results to the cloud. And if we could develop something like this, it would be really powerful, you know, to link the diagnostics and the results to a database to track the infections. So yeah, I agree. It would be really, really useful.
These are fascinating topics. But now I saw a documentary about a dog that is being trained to sniff out COVID. Would this negate all this testing if we could just get a set of dogs to line up and people come along, they have a quick sniff, you've got COVID, sorry, you've had it. I mean, is that even a viable option as a testing method?
I think SAHPRA is going to have to get a canine unit if that's the case, because they have to regulate the dog's performance.
But I mean, is that even a plausible method? Because we know that dogs can sniff certain types of cancer. They've shown in results that is possible. We know that they can sniff various types of drugs at the airports, and they actually make use of that system. Would they be able to actually sniff if someone has COVID-19? I see Manfred's got his hand up. He's definitely want to answer that one. Let me see. Are you unmuted?
You'll have to.
Manfred's not.
You'll have to bring the veterinary sciences in as well to make lots of babies from that dog, because we'll now have to ship them at low cost everywhere. So see that won't happen. So I would rather go for the low cost diagnostic devices. And may I just add that what we also need to look at is a collaboration between industry, then the research community, and also bodies like SAHPRA and the Medical Research Council and so on, that when we start making diagnostic devices in this country and taking research to the industry, that we all work together, that it works out to the benefit of everybody. Then we don't have to start a puppy manufacturing plant, but we can make devices.
Well, I mean, on that note, you actually raise a very interesting point, and that is the South African Medical Research Council and the DSI and the Technology Innovation Agency. They've recently had an open call for grant applications to generate COVID-19 diagnostic tests. And there is currently an open call for grants through the National Research Foundation. And the emphasis is on tests that can be rapidly commercialized. So does COVID-19 offer any unique opportunities for scientists in South Africa to work together or differently in terms of researching and making diagnostic tests that can be commercialized? So I don't know who that would be addressed to. Maybe Kevin, maybe Andrea?
I'll give it a first stab as well. And then I'll go back to the sniffing. Can I also answer something on the sniffing?
Yeah.
There are, just out of interest, there are quite a few research groups, and I know of at least one in South Africa, that are looking at doing breath-based tests. So where you would just breathe into a device, a lot like some sort of a breathalyzer test for blood alcohol. Mm-hmm. They are looking at what's called VOCs, volatile organic components in your breath, that can specifically then diagnose for something like COVID. So I know there's a group at Wits University, for instance, and I'm possibly aware of one other group in South Africa, but internationally there are quite a few groups looking at that. So it certainly is feasible. On the device side of it, I think there's I think there's a really good point to be made that we should be looking at a dedicated program in South Africa for devices, whatever that means, from the reagent side all the way through to getting to the point where we have commercial products. Many of the funding schemes that we have allow for a little bit of R&D or a little bit of development on one aspect, but it really doesn't complete a full pipeline in the South African context. For that, you need, I think, a more substantial program. And the one that Manfred and myself are involved in will play a role in a portion of that pipeline. But I think it's good that we could have a dialogue looking at the entire pipeline and what would be necessary to actually fill that. Because we will get to a time where we can't import any components if there is another pandemic and it is really serious. That is one of our major testing problems now, is we just cannot import sufficient reagents for the tests we have.
Okay, now Andrea, you had your hand up. You wanted to comment?
Yeah, so I just wanted to add with regards to the sniffer dogs, because, you know, if that all pans out, it could be really useful for screening. And then, you know, similarly the way with a rapid test kit, you'd have a confirmatory, you know, lab-based test. So maybe it will be a nice way to just do some screening so that you don't have such a heavy load on the labs to do, you know, the molecular tests.
Labs, as in laboratory, not the Labradors. I'm just checking.
What type of dog are we talking about?
Steve, you're on fire today.
Okay, we've got a couple more questions I want to try and squeeze in if I can. Griselda wanted to know, when do you think we will have access to the Lateral device in South Africa? And what do you think the percentage of accuracy is? It says sometimes pregnancy tests aren't accurate and a urine test is required for confirmation. So could the lateral flow test be misleading?
Can I talk to this one? Yeah. So I don't know, you might have also remembered the scandal when a pregnancy test was testing positive when they poured rooibos on it. So, you know, there are these risks involved. Mm-hmm. If you didn't catch that one.
No, I've been drinking a lot of rooibos, I'm getting worried now.
So, you know, the thing is, yes, we definitely see the possibilities, you know, opportunities that a lateral flow test can bring. But what's important to also note is that everybody's saying, I need this test, I want this test. This is not going to be a valuable test to use when, for example, employers to screen employees every single day. You know, that's not how these tests are developed. That's not the purpose of their use. These things should not be used to facilitate like an immunity passport. So, okay, today I'm negative and now I can go to the cinema or the gym. You know, that's not the place for these tests, because when it comes to accuracy, the current specification is that we want at least 85% sensitivity and 98% specificity. So if we're not getting tests that are performing according to those specs, then we obviously won't allow them to be used. Because again, if you've got a false negative, so it says you're negative, but you actually are not negative, it means you're going to go and infect everybody. If you get false positives, imagine a big company with 1,000 people and half of them test positive today. So now everybody go home and isolate for 2 weeks. But meanwhile, you know, you're not actually positive. So there's a lot of risk involved in having a poor performing test being used. And we also recognize that when a test is authorized for use, everybody's going to want to use it. And it's so critical that these tests are used in the appropriate manner. These point of care tests are for sero surveys. It's when you've got hotspots that you want to identify hotspots. It could inform decision making for government. That's where these things are used. It's not for you to check, you know, my child went to school today. I'm going to test this one to see if he's going tomorrow. That's not the point. That's not how these tests should be used. So yeah, I think that's really just a key message. The performance of these tests is critical. The way that they use is going to be very tightly controlled and prescribed when we do get one that's performing well.
Okay, now we've apparently reached our time limit, but I'm really enjoying this discussion. So I'm going to throw in one or two more questions, and I hope that you guys have got a minute or two just to stick around. Iwazikazi says that there is a group in Cape Town that apparently developed a qPCR testing kit that would produce results in 65 minutes. Is there any news results on that? And is there going to be any rollout? Maybe Tamlyn, you would know?
Yes, so it's actually quite a funny story because there's a company called Cape Bio who have developed these qPCR test kits that I think they are referring to. And then we're Bio Pharms on the other side. So I do know that Cape Bio have developed these tests. I'm not quite sure how far they are in the regulatory process, but I do know that they have done that and they've done an amazing job. So
interesting. So now we are running short on time. So I thought maybe, is there a parting message that maybe each of you would like to give us in terms of things that we should know as the general public with regards to COVID testing? Something that, an important message you want to send out to the people that are participating. And I'm going to start with Manfred. We're going to ask you to unmute.
Yeah, so the message from me is not just on COVID. It's again coming to the establishment of units, and that will help research get into industry. So I invite everybody that has logged into this to maybe contact us. My email address is mrscriba@csir.co.za if you want more information, because we need to work together to make this happen.
We'll certainly post your contact details. We'll get Ryan to sort that out as well. Kevin, would you like to put on a parting message there?
Yeah, I can. Also not directly connected to COVID, because I think I'm talking to the same program that Manfred is. But I just think there are some really, really exciting technologies that we can utilize that can make very specific impact in South Africa. And many of these technologies are being developed to an advanced state internationally on very big budgets, but for different applications. And I think we can leverage and partner with some of those companies to bring those technologies to South Africa, but to solve issues that we have very specifically in South Africa. And healthcare, for sure, is one of those areas.
I mean, look, we've got South Africans that are building rockets and flying Americans to the International Space Station. There's no reason why we certainly can't develop our own local technology here in South Africa. Tamlyn, what would you like to say?
So I guess my message would also be that this country, we have so much talent and so many amazing innovations being developed, and we've got such a strong science and technology sector. And I think COVID has almost been a bit of a fire under all of us to kind of connect and work together. And I think when we do that as South Africans, we are unstoppable. So I'd also like to just, kind of promote the young listeners to study science and get involved, and particularly to also possibly study some business management, because biotech, as I've learned, requires quite a bit of a business acumen. And, we've got so many amazing technologies at our universities that need to be commercialized. And I think there's a lot of strength in that. So yeah, basically science is important and we need to work together.
Excellent. And then, Andrea, before you answer, there was one thing that's been bugging me, and that is, if I do come up with a test and I'm working at a university, who owns the intellectual property to that? And then you can give us your message.
That one I don't have the answer for. Maybe the profs or Dr. Manfred can pronounce on that one.
Okay, we'll give. You're going to give your message. We're going to quickly go to Dr. Manfred just to ask him if he's got a response to that one.
Yeah, so you asking if somebody develops something at the university, who owns the intellectual property?
Do I own it as a postgrad student? Does the university own it because I'm part of that university?
Yeah, the university owns it, but you are the inventor. So if you file a patent, you are the inventor on the patent. And there's an IPR Act that government has in place whereby you share also if there's profit from that invention. We need a bit more time. There's not enough now, but if you're employed by the university or if you're a student, you sign an agreement that they own the technology.
I see. Aha, so you have to wait until you actually graduate. And then, Prof, you wanted to add to that? Janice?
Oh, yeah. And Steve, so that's actually quite important. And, you know, with our research, a lot of it is publicly funded. And so it makes sense that, you know, the university is a public entity and therefore it owns it. And then working with organizations such as NIPMO, we then can, you know, if there are any benefits, you know, from the products. But I think it's quite important, this matter of intellectual property. So inventors do have a place, and it doesn't just go somewhere else. But the most important thing about the IPR Act, I feel, is that the benefits ultimately accrue back to South Africa.
Awesome, awesome. And then, Andrea, you get to have the final word. What is your parting message?
Thanks so much. So the song I want to sing is that when we've got, when SAHPRA does authorize test kits, whether they are molecular ones or serology test kits, we do place the listings on our website. So please, you can always check, especially with the serology test kits, because I do fear that there are some that were illegally imported and so on. If you've got, you know, if you've seen any test kits anywhere, you can always, and I'm saying for future when we do list these, please check on the website to make sure that whatever's being used has been authorized for use by SAHPRA. If it hasn't, please don't use it, because we cannot guarantee the performance of the test. So your test results might not be an accurate result. And then also just to report any, you know, advertising or website sales or sales on social media of these type of test kits are in contravention of the Act and should be reported. Thanks.
Thank you, thank you. Now, before I hand over to Janice to say a final thing, we just want to remind you that our next webinars for the Scifest webinars are taking place on Friday the 19th of June and Tuesday the 23rd of June. The topics and the speakers will be announced shortly. You can visit the various social media platforms for Scifest Africa. And if you have any information, you can send an email to info@scifest.org.za. That is info@scifest.org.za. And Janice, I hand over to you to sign off.
Okay, great. Thanks so much, Steve. And thanks everybody for coming together for this panel and this discussion. I think for me personally, the great thing as a scientist that I've seen the last couple of months is that there is a lot of expertise in South Africa, and that putting together, we actually probably going to see some direct benefits for the biotech sector and product development. So the work that Tamlyn is doing, for example, in developing new reagents, will drive the prices down, because at the moment we import everything from elsewhere. So having a local supply is going to be very important, not just for research in diagnostics, but research more generally. So I think that there will be a positive spin-off for biotech and for research in general in South Africa across the board. So thank you, Manfred. Thank you, Kevin. Thank you, Andrea, for all of your hard work in this sector. And finally, Steve, thank you. You've been a wonderful host. And great. Thank you, everybody.
Thank you, everyone. We'll see you at the next webinar. Bye-bye.